In the ongoing lecture series on plant tissue culture, in the previous few lectures, we started with the historical development of plant tissue culture technique. Then we discussed about the ideal characteristics of a plant tissue culture laboratory or the features of a plant tissue culture laboratory. And in that I told you that plant tissue culture laboratory is not a single laboratory but a complex of small laboratories. And in that we also discuss what are the different small rooms or laboratories that are required with respect to the infrastructural requirements as well as the equipment that is required in each smaller laboratory or each room depending upon its use. Then we discuss about the steps that are involved in the plant tissue culture experiment, starting from the cleaning and sterilization of the glass apparatus, followed by the sterilization of the plant material, establishment of the culture, sterilization of the culture media, preparation of the media, what are the various components of a culture media, what is the function of those ingredients of the culture media. After that we discussed about the types of plant tissue cultures, and we broadly categorize them into organized and unorganized, and in unorganized we discuss about the culture and suspension plant tissue cultures, and under organized class we discuss about the cultures that are established using different types of plant organs, like root culture is there, anther or pollen culture is there, or protoplast culture is there, and so on and so forth. Then we discussed callus culture in detail, that what is callus, how the culture is established, what are the applications of the callus culture. So friends, in continuation with the lecture series, today I am going to start with the suspension culture. Now, as the name suspension indicates, this type of culture is largely in the liquid form, or we can say the media which is used to establish the culture is in the liquid form. And the suspension cultures can be initiated through the cells that are obtained from callus. As we discussed previously, that callus is nothing but the undifferentiated mass of the cells that is grown on the culture media, solidified culture media. We can use that callus, we can separate those cells, and we can suspend those cells in a liquid media, thereby we are preparing a suspension culture. Now, the basic difference between the media that is prepared for callus culture and suspension culture is the absence of solidifying or the gelling agent in suspension culture media. I hope you remember that we discussed that certain gelling agents or solidifying agents are used, such as agar is there, agarose is there, which are used to solidify the culture media, to gellify the culture media, and used for the growth of the callus. But in case of suspension culture, that gelling component, solidifying component is absent. Rest of the composition of the culture media is by and large same. Again, it will depend upon the type of the plant you are using, type of the initiating cells you are using. So, in case of suspension culture, we can initiate by using the cells obtained from callus. We first disintegrate that callus into smaller groups of the cells, or preferably single cells, and then we suspend them into the liquid media. Or we can directly use the explant material. Say for example, if you want to develop a suspension culture from mesophyll cells of a leaf, we will first take the leaf, we will cut it into smaller parts or small pieces, then we can separate the cells by using mechanical or enzymatic method. Obviously, mechanical method leads to certain mechanical damage to the cells, thereby decreasing the number of the viable cells, and therefore this is the major drawback of mechanical method of obtaining cells from the explant. Therefore, since the viability is very less, it's very difficult to maintain such kind of suspension cultures. On the other hand, the enzymatic method, we use specific enzymes which dissolves the middle lamella or the area where the two cells join, because the natural cells have a tendency to adhere to each other, and these enzymatic solutions basically which contain specific enzymes that dissolves the middle lamella of the two cells and separate those cells. So once we separate the cells from the explant, we can directly use them to the development of a suspension culture. Now, as we discussed that suspension cultures largely occur in the liquid media, and therefore the liquid media has to be continuously agitated during the culturing. Now, what is the role of agitation of this liquid media? Now, the most important role of agitation is it helps in preventing the formation of cellular aggregates or the clumps. As I told you that the cells have a natural tendency to adhere to each other, such kind of agitation or stirring of the liquid media will help in preventing the formation of those aggregates. Further, this type of stirring also helps in breaking the callus, though we first disintegrate the callus into smaller pieces and then we suspend them into the liquid media, but sometimes we directly use the callus and suspend it into the culture liquid media, and this kind of stirring or agitation helps in breaking that callus into smaller fragments or preferably into the single cells, thereby helping in proper maintenance of the suspension culture. Agitation or stirring also helps in maintaining the uniform cellular density in the culture media. Now, this is important because if the cellular density in the media is not uniform, obviously then in that particular, suppose in a particular area the cellular density is very high, then in that particular area the exhaustion of the culture media will be quite rapid in comparison to the other areas of the culture vessel, and again this will cause the non-uniform growth of the cells, and therefore stirring or agitation always help in maintaining the uniform cellular density across the culture vessel. And last but not the least, it also helps in the proper aeration or the circulation of the gases across the liquid culture media. Now, what are the various factors that are responsible for the aggregation or clumping of the cells in the liquid media? First is the plant variety. Now, it has been seen that there are certain plants where or whose cells have a natural tendency of getting into aggregates, or the tendency of getting into aggregates is higher in some plants such as Nicotiana tabacum, and it has been observed that the cells of Nicotiana tabacum has a higher tendency to form aggregates, whereas there are certain plants like Mirinda citrifolia whose cells have a natural tendency to remain separate from each other, or the tendency of forming aggregates is far less in comparison to Nicotiana tabacum. So, depending upon the plant variety, aggregation will vary. Another factor is the culture media. The composition of the culture media sometimes also helps in preventing the aggregation or clumping of the cells. It has been observed that the media which are having higher oxanic concentration or oxen concentration, they have a lesser tendency of aggregation. The reason is oxens have been found to stimulate the enzymes which causes the solubility of the middle lamella or the cell wall components which are responsible for the cellular aggregation, and thereby they decrease the aggregation. Third is speed of agitation. Now, as we discussed previously that agitation or stirring of the liquid media always helps in preventing the aggregation, or it helps in breaking the clumps of the cell. So, if the speed of agitation is not proper or is very slow, it will allow the cells to get into close proximity of each other and they will form the aggregates. On the other hand, if the speed of agitation is very high, it may cause substantial damage to the cellular morphology. Therefore, speed of agitation has to be very optimum and has to be optimized for each type of the culture. Last but not least is the growth phase of the culture. Now, as we discussed that during in the callus culture, there was a phase in the growth pattern of the callus culture, that is the exponential or the linear phase, wherein there is the highest rate of cell division. So, as the rate of cell division will be higher, larger number of cells is formed, higher the number of cells, more is the possibility of aggregate formation. And therefore, it also depends, aggregation also depends upon the growth phase of the culture. So, depending upon the growth phase, we have to optimize the other conditions during the culture. So, we can say that if we look into the overall composition of the suspension culture, we will find two types of cellular fragments, or we can classify the components of a suspension culture in two categories. First category will be of the free cells. Obviously, these cells will be of different sizes depending upon the growth phase in which they are. And the second category will be of the cellular aggregates, which consist of 2 or up to 10 cells or even more. And in such type of aggregates, it has been found that the smaller cells are present largely in the central region of the aggregate, whereas the size of the cells increases as we move towards the periphery of those aggregates. And because of such kind of observation, it is hypothesized that the cellular division is highest within the center of the aggregates, and from there the growing cells proliferate towards the outer side. If we look into the growth curve of the suspension cultures, we discuss about different phases of growth in callus cultures. Similar kind of 5 phases you can see in the growth curve of the suspension culture, and these 5 phases are lag phase, exponential phase, linear phase, diminishing growth phase and stationary phase. Now, lag phase is somewhat similar to that of the induction phase of the callus culture, or in simple words, if I say, in lag phase, cells prepare themselves or acclimatize themselves to the chemical environment of the media in which they are suspended, and they prepare themselves to divide. In the exponential phase, now the cells have become acclimatized to the chemical environment of the culture and they start dividing, and this division becomes more rapid in the linear phase, or we can say that there is a linear growth of the cell division with respect to time in the linear phase. We can also say that maximum cell division during the culture is observed in exponential and the linear phase. The fourth growth phase is the diminishing growth. Now, once the cells have flourished very fast in the linear phase, they are utilizing the nutrients present in the culture media with the same pace, and therefore the media starts getting exhausted after the linear phase, and this exhaustion of the media causes the occurrence of this diminishing growth phase, wherein the cell divisions get slowed down because the nutrients have been exhausted during the linear and the exponential phase. Now the cell growth becomes slow. And last is the stationary phase, wherein the media gets completely exhausted and the cells stop dividing, and if the exhausted media is not immediately replaced with the fresh media, these dividing cells will die. Therefore, it is always advisable to replenish the media with the fresh media before attaining the stationary phase. Now, we move on to the types of suspension cultures. Now, suspension cultures, depending upon how the fresh media is introduced, we can categorize them into batch cultures and continuous cultures. Now, how the media is replenished, on that basis, continuous cultures can further be categorized into closed continuous cultures and open continuous cultures. Open continuous cultures are further divided into two types, turbidostat and chemostat. There is a third type of suspension culture, which is synchronous cultures. Now, in synchronous cultures, as the name indicates synchronous, it means that we try to separate the cells on the basis of their position in a growth cycle. Suppose certain cells are growing in M1 phase, certain cells are growing in M2 phase of the cell cycle. So, we will try to separate M1 cells and M2 cells, and we try to culture these cells separately so that we get a synchronous growth of the culture. Now, we will discuss the different types of suspension cultures in detail. First one is batch cultures. Now, as the name batch indicates, in such kind of cultures, once the media is exhausted or near exhaustion, then the complete culture or culture media is replenished by the fresh media. So, what is done in batch culture is, we take the cell suspensions are normally grown in 100 to 250 ml flask, which contain around 20 to 75 ml of culture media. So, once the media is near exhaustion, we take out small volume of that suspension, and that suspension, small amount of suspension containing growing cells, that is placed in a fresh media which is around 5 times its volume. So, for example, if we are taking 1 ml of the suspension culture, we will dilute it with the 5 ml of the fresh media. So, this is how batch culturing is done. Now, such kind of batch culturing is largely used for initiating single cell cultures. Now, obviously, there are certain advantages of batch cultures. Now, the cells that are present in batch cultures, we can always see all the 5 stages of the sigmoid growth curve of the suspension cultures, that is lag phase, exponential phase, linear phase, diminishing and stationary phase. So, if you want to study the physiology of the cells in different phases, growth phases of the sigmoid curve, then batch cultures are the best technique. We can always reduce the lag phase in such type of cultures. Now, this can be done by introducing the conditioned media. Now, conditioned media is nothing but the media wherein the cells were previously dividing. Say for example, we take small amount of the media when the cells were there in the exponential or the linear phase, and we introduce that media into the fresh media. Now, this will help in early acclimatization of the cells, because they will find similar type of culture chemistry which was present earlier or to which they were exposed earlier, and therefore, this helps in reducing the lag phase. The cells easily get acclimatized and they start dividing. We can also maintain such kind of batch cultures in exponential phase if you want to. This can be done by frequent subculturing or by frequent replacing the exhausted media with the fresh media before it enters into diminishing phase. Now, this always helps in maintaining the cell division in the exponential phase. There are certain disadvantages also, since we are, if you want to maintain them into exponential phase, we are constantly changing the media, we are constantly subculturing the culture. By this, there is a constant change in the cell growth and metabolism pattern of the cells, and since we are very frequently replacing the fresh media or the spent media with the fresh media, there is slight change in the media composition. Now, all these factors leads to the absence of steady state growth of this type of culture, and since there is the absence of a steady state of growth, therefore, such type of cultures are not found suitable if you want to study the pattern of a cell growth or the metabolic pattern of the cell. Now, we move on to the continuous cultures. As the name continuous indicates, here the exhausted media is continuously replaced by the fresh media. In batch cultures, wait for the media to get exhausted and then we replaced it with the fresh media. Here the fresh media continuously replaces the media at a constant rate. So we have closed and open continuous cultures. Now closed cultures, in closed cultures or closed continuous cultures, the addition of fresh medium is balanced by the outflow of the old media. Say for example, we have adjusted a outflow of 5 ml per minute. Then in such type of continuous culture, fresh media will be added at a rate of 5 ml per minute so that a constant balance of media is maintained. Whereas in case of open continuous culture, addition of fresh media is balanced by harvest, which also includes the cells, and this helps in maintaining a constant submaximal growth rate. Here submaximal word is important because we do not allow the cells to reach to the maximum growth rate by continuously replacing the media. And how this is maintained? Depending upon the mechanism by which this constant composition of the media or constant rate of media is maintained, we have chemostat open continuous cultures and turbidostat open continuous culture. In case of chemostat open continuous culture, again as the name chemostat indicates, we maintain a static chemical environment of the media. In this, a steady state of cell growth is maintained by constant inflow of fresh media in which the constituent which is responsible for the growth, say for example nitrogen is responsible or phosphorus is responsible for the cell divisions of the cell or the growth of the cells. So we keep the concentration of nitrogen or phosphorus towards the lower side in comparison to the other constituent whose concentration is always higher than the required concentration. Therefore, the nitrogen or phosphorus will become a growth limiting constituent, and therefore, since we are limiting their concentration, we are allowing the cells to achieve a constant growth rate, and this is how chemostat open continuous cultures work. But there are certain problems in chemostat cultures. First one is the foaming. Whenever fresh media is added, since it is continuously added, it comes in contact with the exhausted media into the culture vessels. It has been observed small amount of foaming occurs, and this kind of foaming sometimes traps the growing cells in it, thereby breaking the static nature of the culture vessel. To avoid such kind of foaming, anti-foaming agents are now used in those culture vessels or media, which always help in maintaining the uniformity of the chemical environment in the culture media. Then, since agitation is going on in these kind of, as you can see in the diagram, there is a pedal rotator is there. So this kind of agitation or stirring is there in chemostat type of open cultures. Such kind of agitation can cause damage to the fragile cells. So in order to overcome such problem, the rate of agitation or stirring is always optimized before starting the culture. So gentle agitation can be done. Further, even the aeration, if the rate of aeration or sparging the air into the culture vessel is very high, that aeration or the pressure of the gas can also damage the cells. Therefore, we can always control the rate of aeration as well. Further, it has also been found that certain cells get adhered to the walls of the culture vessel and they start growing there only, which sometimes also lead to certain bacterial infections in the media. This can be overcome by giving a saline treatment or saline treatment to the walls of the culture vessel, which make them hydrophobic, thereby preventing the adherence of the cells to the walls. So these are few problems which can be overcome. So in order to maintain a static environment in chemostat, we have to go for a gentle shaking, which should not upset the uniform chemical environment in the vessel. Further, It has also been found that whenever media, fresh media is added, a small pulse in the media is generated. Then this kind of pulse also sometimes upset the static property of the chemostat. So therefore, the addition of the media has to be very gentle and in a very optimized manner. Second is the turbidostat. Now this was introduced by Bryson and Sibilaski in 1952. In such kind of continuous cultures, the flow of the fresh media is controlled by the turbidity of the culture media, or we can say that the turbidity in the culture media largely governs the flow of the fresh media. So here you can clearly see in the diagram, this turbidity is determined by the photocell, which is placed in the near vicinity of the culture vessel. So this photocell determines or measures the optic density of the culture media, and it sends the signals towards the valve sensors. So if the turbidity or the optical density or the turbidity of the culture media increases, it sends signal to the sensor of the fresh media valve. The valve gets open and the fresh media enters into the culture vessel, thereby maintaining the required turbidity. Because we know that as there is growth of the cells, the number of the cells or the cellular density increases. So as the cellular density increases, the turbidity in the culture media also increases. Now the turbidity in the culture media can also be maintained by flushing out certain volume of the cells. So this is how turbidostat works, by maintaining the turbidity of the culture media. Now there are certain limitations of turbidostat continuous cultures as well. It has been found that on the optical surfaces of the detector, sometimes the foam of media gets deposited or certain precipitates get deposited over a period of time. Some microbial growth occurs on the optical surfaces of the photocells. Now such kind of hindrances on the optical surface of the detector will give you the false reading about the turbidity of the culture media, and therefore it becomes highly unreliable to maintain those cultures on the basis of turbidity, and therefore it is always required a regular maintenance of those detectors. So friends, this was about the continuous cultures. In the next lecture, we will discuss about certain large scale continuous cultures. Thank you.